very interested in using transgenic strategies as a way of breeding better barley cultivars, and in that context, the unintended changes that can occur with transgenics are undesirable to me. And so today, I've got a presentation for you to talk about the way that we are dealing with unintended changes that occur during the transformation process of barley, both from the standpoint of avoiding them with our clean gene delivery system, as well as removing some of these unintended changes with some breeding strategies that we employ. I work for the USDA ARS in Idaho. I just included this slide at the beginning of my presentation to remind myself that my real love and passion is in application. This is a field of non-transgenic barley, a variety that I developed. And I'm always thinking in terms of developing materials that can succeed in a commercial agricultural setting. And that's the viewpoint that I'm going to bring to the presentation and the viewpoint that I bring to my work. So I'm obviously interested in creating good transgenic barley, and by good I'm talking about achieving what the dream of transgenic barley always was, or any transgenic crop, which is to be able to make limited and predictable changes, which are essentially improvements. We want to add, for instance, a trait like Fusarium head blight resistance, but we don't want to alter the genetic package or background that we're making that change to. We don't want to interfere with yield or malting quality or other traits that make our barley cultivars useful to producers and to industry. If we have these unpredictable and undesirable changes, they essentially become impediments to commercial use. And I have a couple of ways of dealing with these things. I can either prevent the problems from occurring in the first place, or after they occur, we can fix the problems, at least to a certain extent. So one of the first things that we ran into when we started working with tissue culture systems relative to barley was we ran into somaclonal variation. I guess I would call somaclonal variation the original unintended effect. And it occurs as a result of tissue culture, and it consists of multiple genetic and epigenetic changes that occur as a result of passage through tissue culture. Somaclonal variation is widespread. It affects many loci across the genome. And back in the day, when mutation breeding was all the rage, somaclonal variation was thought of as just another mutagen, a way to induce new and useful phenotypes in much the same way that people used radiation or chemical mutagenesis. And there was some application of this, but by and large what we've been using tissue culture for is for the production of transgenics. And for transgenics, somaclonal variation is a menace, not a benefit. And I say this because we have found with barley that simply passing the tissues through tissue culture affects the agronomic and malting quality of the derived lines. And of course the effects are not positive, they're negative. If you think about all this random variation occurring in a genome, it's kind of like throwing a wrench into a functioning piece of machinery. It's going to damage the gears, not polish them more than likely. So the somaclonal variation, which we are not in total control of, creates negative performance characteristics. And just to highlight a couple of them, from these studies that we looked at the performance of the barley lines in, we saw that 26 of the 30 lines that were studied, and these lines came from 6 different cultivars, 26 of the 30 lines were changed for at least one agronomic trait. Mean yield reductions were seen of 2 to 12%, which some of you might not think of as being all that negative, but really it's in the wrong direction. And farmers are pretty sensitive to several percentage points of reduced yield, and if we give an FHB-resistant barley to a farmer, one of the things he or she is going to be concerned with is whether or not it performs well for traits other than the Fusarium head blight resistance. And one of these important traits to a barley producer is the malting quality of that line, because that's where, especially in the States, where the big money in barley production is. And this package of biochemical characteristics that make up malt quality is something that the maltsters and brewers have very little tolerance for deviations from. And so when we saw the agronomic alterations in these lines, we also saw alterations in malt quality. And so this said to us that one of the things that we were going to have to do in producing transgenic barley plants was deal with this somaclonal variation. Now, at the time of these studies, transgenic barley had not yet been produced. So this is just what we see by passing the materials through tissue culture. When Peggy Lameau at the University of California successfully produced the first transgenic barley lines, I tested these. And what we saw, unfortunately, was that somaclonal variation in transgenic plants was actually exacerbated compared to non-transgenic plants. The first work that was done to look at this was a field study. I took some of the original transgenic parents that were hemizygous, meaning they were segregating for the transgene, and we derived some non-transgenic or null segregants from these transgenic plants, and then increased them and then put them out in the field and looked at their performance. And what we saw was that the yield reductions ranged from 15 up to 84% in these non-transgenic derivatives of transgenic plants. And to reiterate, these were not transgenic plants. We're looking at somaclonal variation here causing this problem, not the insertion of the transgene or the expression of the transgene. Well, needless to say, this data didn't please us very much, and my colleague Peggy, when she saw this data, decided to have some of her laboratory personnel look into this issue a bit further, and there were a couple of studies published that looked at the cytology of transgenic plants compared to non-transgenic plants. And what was seen was that transgenic barley plants had more cytological variations than non-transgenic plants. And there were specific in vitro conditions that were used in the transformation process that were implicated in producing this additional variability. And for instance, the plasmolysis, or the partial plasmolysis step that is applied to the immature embryo explants prior to bombardment, that was associated with greater variability, as was the imposition of a chemical selection agent. And of course, both of these things were just part of the transformation process. And workarounds are not necessarily possible around all of these kinds of things. They just go with the technique that we're using. So what can we do about this kind of a thing? Well, backcrossing is a technique that is capable of removing unlinked and unintended changes. So if you think about our transgenic plant here that I've got depicted as a blue genome here, and the little yellow star represents the position of the transgene that we're interested in. If you think of this blue genome as being chock full of somaclonal variation, there's just many, many different places in the genome that have a change that we want to get rid of. Backcrossing is a way to basically transfer that gene into another background and discard the background that's full of somaclonal variation. So just a quick schematic representation of what backcrossing is from a quantitative genetic perspective. You start with a transgenic donor parent and cross it to a non-transgenic recurrent parent. The recurrent parent could be the original genotype that you achieved the transformation in, or it could be another genotype. But the process is to make a cross, and in the progeny, the population as a whole will have 50% of the donor parent genome replaced by the recurrent parent genome. And as long as you select for the transgene itself, the population will be improved with respect to being transgenic, but also having less somaclonal variation. And so this process can be repeated, and at each cycle of crossing, you're getting rid of another 50% of the remaining donor parent genome, so that after the first back cross, which is the second cross, but the first back cross, we're now down to 25% donor parent genome, and with successive crosses, you continue to reduce the percentage of the genome that is associated with the donor parent. And so this is just showing a few back crosses. It's not what we would consider a true isogenic line, but if you would go a few more cycles, you can get this down to, you know, where you have less than 1% of the original genome left, but you will still have your transgene and would presumably still have your transgenically encoded traits. So this is the theory. What happens with practice? In practice, it worked for us. In this particular study, we looked at a number of lines, transgenic barley lines that were in the cultivar conlin background. On average, these lines had a 31% yield reduction. This is just coming straight out of tissue culture and being self-pollinated and advanced to have enough seed to do a yield trial. But we also did 2 crosses back to the wild-type conlin plant that had not been through tissue culture, and we saw in the derived lines that the yield reduction was just 6%. So this was pretty encouraging to us. And so it's one way, it's a time-consuming way of getting rid of somaclonal variation, but it's an effective way of getting rid of it. The reason that we can get rid of it again is because it's unlinked to the transgene itself. But what happens if we have an undesirable characteristic that goes along with the transgene itself? We can't get rid of the transgene without getting rid of our trait. So the kinds of characteristics that we're addressing, hopefully with the technique that we're using in our laboratory, is there's three things that I've put down here. The first of all has to do with gene expression. Often when transgenes go in, they become rearranged. Some of these rearrangements result in inverted repeats that can cause double-stranded RNA to be formed, which is a potent trigger of RNA-induced transgene silencing. So we think if we can avoid this, we can get better expression characteristics. We would also like to address the accompanying baggage with transgenes that is typical. Typically, you have a selectable marker that accompanies the transgene, which we use in vitro, but we don't need it in the final product. And we also typically have bacterial vector sequences derived from the cloning vector that are incorporated into that transgenic locus. Neither one of these last 2 features are necessary, and especially from a consumer acceptance point of view, they're somewhat undesirable, and we would like them not to be in our final product. So how can we prevent unintended changes? Well, I first talked to you about somaclonal variation as just something that goes along with tissue culture. This is true, but we've also found that we can moderate the somaclonal variation by our culture methodology. There's less somaclonal variation associated with highly differentiated meristematic tissues we have discovered. So we use a particular auxin-cytokinin ratio in our culture media to drive our cultures towards this highly differentiated state. And also we minimize the selection pressure that we use by using the lowest concentration and keeping it in with the culture media for the minimum time necessary for selecting the transgenic tissue. We use agrobacterium transgene delivery instead of biolistic gene delivery because we have better odds of having an intact single locus when we use agrobacterium. And then finally, we are striving to deliver a clean transgene, one that has no rearrangements, that can invoke gene silencing, no selectable markers, and no vector DNA. We're doing this with a process of gene delivery via transposition. And I think many of you will know what transposable elements are, but in brief, transposable elements are small pieces of DNA that are mobile in the genome. They can move from one place to another. The particular system that we are using is based on the Mays ACDS system. This is a very famous family of transposable elements. It was first described and researched as far back as the 30s. This is what Barbara McClintock won her Nobel Prize for. And so it's been exhaustively and intensively researched. There's quite a bit known about how it acts and how to exploit it to do things such as moving transgenes around. In this system, there's an enzyme called AC transposase. This can interact with short terminal sequences that flank that are on, well, short termini. These sequences are on the terminal regions of the AC and DS elements. And when AC transposase interacts with that, it can these sequences, it can cause either the AC or the DS elements to move from one location or another. DS elements differ from AC elements primarily because they don't have a functional AC transposase in them. Other than that, they have the same terminal sequences that allow them to move. And because there's a lot of variability permitted in a DS element, we can produce synthetic DS elements by bordering transgenes with short DS termini, and then these transgenes can be used as a transposable element, basically, and moved about the genome. And we have found that transposed loci that are, when they go into their final location, we find them in gene-rich areas, and we find that the gene that's being moved around has high inheritable levels of expression, which is exactly what I want as a plant breeder. I want my obviously, not only do I want to make limited and predictable changes, but that predictable change that is limited, I want it to stay there. I want whatever my transgene is to keep functioning. Schematically, the system looks like this. It's a two-step process. The first step is to produce an initial transgenic plant. The transgenic locus in this plant is pretty typical. It has a gene of interest. It has a selectable marker, and these asterisks that are on the that I've got depicted on this locus are meant to represent things like vector sequence, rearrangements, and other things that are undesirable. I like to use the technical term garbage for all of this. But there's one difference from a typical transgenic locus, and that is that our gene of interest is bordered by these DS terminal sequences. The second step is to cross these plants with a plant-expressing AC transposase, which can cause this DS-bordered gene of interest, or the synthetic transposon, if you will, to jump out of its original location and to reinsert in some other place in the genome. It can theoretically jump to almost anywhere on any of the other seven chromosomes. And so we get these genes of interest now are delinked from our original site, and all that garbage can be left behind by genetic segregation, and we can identify plants that have our gene of interest only without all the rest of the garbage that came along with the original transgenic locus. So, how can this contribute? How can delivery by transposition contribute to predictability? First of all, we get intact transgenes with distinct borders, which facilitates sequence-based transgene mapping. We should reduce or eliminate expression that is compromised by rearrangements. And we also remove the potential for pleiotropic effects of the selectable markers or other unnecessary sequences that are in there. We're also using transposon-based delivery to efficiently produce populations that have intact site-specific recombination platforms. We're going to try and move these site-specific recombination platforms to a variety of sites within the genome, and then select lines that have these sites in areas that support appropriate transgene expression, and also that don't have pleiotropic effects. For instance, we would select against one of these platforms that inserted itself into a native gene. And then these kinds of lines could be characterized for their performance and basically form what you might call founder lines, lines that we can use for a variety of experimentation. And so we think this can help us in the future work with our transgenic barley and have much more predictable products from our laboratory work. And then finally, just a note of caution. If you want to avoid unintended effects, don't be surprised by things that you can see with good, solid observation. And here's a problem that we ran into. We attempted to do some mapping of some transposons that were moving around the selectable marker bar. And we were going to do this by creating biparental mapping populations, and these golden promise lines were crossed to a genetic stock called Oregon wolf barley dominant that's used a lot for mapping because it's quite variable. And the problem was that our hybrids died, and if you see on this picture on the right is a normal wild-type plant, and to the left there are progressively more severe phenotypic disturbances in some hybrids. In fact, the one pot on the left, you see these plants that barely have survived. These are all the same age. And what we found as the source of this problem was that this particular line of barley was sensitive to the phosphoenothricin protein that is encoded by BAR. And so this is quite a negative pleiotropic effect. And so this is one of these things that's concerning to me, but we have a system to get rid of the selectable marker, and so this in our system, this is a good example of something that we could avoid with our system. Okay, so finally, I'd just like to acknowledge two long-term collaborators, Peggy Lameau at the University of California at Berkeley, and Lynn Darlene, also with the U.S. Department of Agriculture, but at a different location than for me, Fargo, North Dakota. These two researchers are the ones that until recently were producing all of the transgenic barley that we're talking about. Peggy Lameau worked with the Golden Promise background. Lynn Darlene has been working with the common background. And then recently we have begun doing the transformation work directly in my laboratory as well. Over the years, we've been funded by our respective agencies, the U.S. Department of Agriculture and the University of California. We've also received some funding for some of the materials I talked about today from the National Science Foundation, the U.S. Wheat and Barley Scab Initiative, and from the American Malting Barley Association. Thank you for your attention.